Welcome everybody. Today Mariel is not here with us, but I'm gonna take care of introducing our guest. As you know, I'm Veronica Martinez, and our guest today is Alvaro Alvarado. He's originally from Peru, beautiful, beautiful country. He has a BS in biology from the Universidad Peruana Cayetano Heredia. And then he came to the United States and he performed a PhD in molecular medicine in the Case Western Reserve University, where he was studying glioblastoma. And after he moved to LA, where he became a postdoc, and he's now a project scientist, where he is keeping his study in glioblastoma. He's in the lab of Dr. Kornblum in the Semel Institute for Neuroscience and Human Behavior in the University of California in LA. And today he's gonna talk about reconciling glioblastoma heterogeneity. And so, you know, he's in the market, so if somebody wants to offer him a lab, sure that he's happy about that. Okay, thank you so much, Alvaro, for being here with us, and go ahead, please.
Well, thank you guys for inviting me to the campus colloquium seminar. Today I'm going to talk about targeting heterogeneity in glioblastoma. And thank you for that nice introduction. So today I'll give a brief introduction on glioblastoma, and then I'm going to talk about two projects that shared in common the fact that we are trying to reconcile the difference between the tumors using pathway-based approaches. One is using genetic expression, and the other one is using mass cytometry. And I'll end up with some future directions. So glioblastoma, it's the most common primary and most aggressive brain tumor in adults. It's uniformly fatal, and the median survival is about 15 months, and the 5-year survival of patients is less than 5%. And I've been in this field for about 10 years, and these numbers have not changed since I started studying. So the need for developing new therapies, it's paramount. The normal standard of care is using chemotherapy and radiotherapy. And more recently, there's been efforts to try to develop targeted therapies, as well as looking at the interaction between tumor and immune cells and trying to overcome the immune resistance that could be in there, and developing the immunotherapies that go together with the standard of care. And I will talk about tumor immune interactions in the second project. So what are we talking about when we talk about heterogeneity? There's actually two things. One is intertumoral heterogeneity, so the difference between the different tumors from different patients. And over 10 years ago, there was a nice effort from the Cancer Genome Atlas Consortium trying to identify different molecular subtypes of GBM. And they came up with 4 different molecular subtypes, then narrowed out to 3, mainly proneural, classical, and mesenchymal. And there's different genetic aberrations associated with these molecular subtypes. But after a few years, and with the appearance of single-cell RNA-seq, these studies were further developed into looking at intratumoral heterogeneity, so the difference between different cells within one tumor. And so what they found was that within the same tumor, the presence of these different molecular subtypes was possible. And so therapies that were aimed at targeting one particular subtype will not work because one tumor could present different molecular subtypes. And so together with this intratumoral heterogeneity in terms of molecular subtypes, we also have the existence of these cancer stem cells that have been described in several solid tumor types, including the brain. And so what cancer stem cells are these cells that have some sort of capability of differentiating into different cell types, and also are able to sustain their own self-renewal. They can be characterized by different markers, mainly surface markers. You can see them here, C133, C15, C44, C49F, and so on. So these cells are able to initiate tumors when implanted into mice, and they're able to recapitulate the original tumor heterogeneity of the tumors that we observe in patients. And furthermore, there's also the presence of spatial heterogeneity, where the cells within a tumor are not the same at the core versus the edge of the tumor. And there were some nice studies looking at how different mutations can arise in cells from different regions of the brain. And so in the second project, I also look at some spatial heterogeneity. So with all this in mind, we try to develop a couple projects that sort of leverage this heterogeneity into developing new therapies for the treatment of GBM. And so the first project is pathway-based approach reveals differential sensitivity of GBM to E2F1 inhibition. And this had a very simple idea, was to take the expression data that many people have looked at, and instead of looking at genes themselves, looking at pathways. And the way to do this is to look at genes and enrichment. So genes and enrichment typically compares two samples, and based on the expression of different genes, gives a score for a particular gene set. And then you can compile different gene sets, and you see which ones are different in sample A versus sample B. So what I did here, and what is represented in this graph, is rather than comparing one sample versus another, is I took all the samples that were available in the Cancer Genome Atlas, and I compare each sample versus the average of all the samples. And so each column here is that comparison, and each row here is a particular gene set. And I'll talk about the details in the next slide, but just the overall approach was to then take the scores that I generated here and try to cluster cells based on their pattern of enrichment for these particular gene sets, or for the scores that were generated for the gene sets. And once I had them in a spatial distribution, try to see how each gene set contributed to a particular direction, get the common elements that identify each of the clusters, and then using more bioinformatic tools, such as ingenuity pathway analysis, try to identify some genes that are targetable or some drugs that we can use to validate this approach. So as I said, the first step was to generate genes and enrichment patterns for each of the samples. And this work was recently published in Cancer Research Communications last month. So here you can see the original 4 molecular subtypes from the Cancer Genome Atlas paper. And so what I did was generate these genes and enrichments, and then try to cluster these samples based on their pattern of enrichment. There's 2 different collections that I use. So the GSEA software has different collections based on what you want to look at. The canonical contains canonical pathways that have been described in different cell types. And the oncogenic are things that can be dysregulated in different cancers, or that arise from the overexpression or deletion of particular genes of interest in some cancer models. And so based on this, I generated 3 clusters for the whole Cancer Genome Atlas collection, which is about 550 samples. And so there were 3 clusters that represented based on how the genes and enrichment work in this case. Now, it's important to see that if I take those same samples that I've clustered and I label them instead of the cluster that I identify, if I label them based on the classic molecular subtype, there is no correlation, meaning that the clusters that I identify are not the same as the molecular subtype that were first described. And the second important point here is that the clusters that I identified, when you look at the patient survival in each of these clusters, there are differences between them. And so the clusters that we have identified have some sort of clinical significance that is important. And the original molecular subtypes did not have this characteristic. Another point here is one of the clusters that have the better survival in this case was cluster 1 for this particular collection. And you can see that it's composed equally of classical and proneural samples, meaning that the one subtype is having the better survival as was originally thought. I also did some gene ontology analysis on these samples, and this is just showing some of the gene ontology terms that are enriched in each of the clusters. So in this case, there's a cluster 3, that is enriched for a lot of extracellular stuff and inflammatory processes. There's another cluster that is highly enriching synaptic and secretion of what is normally in neurons and astrocytes. And there's another one that is cluster 2, highly enriched in cell cycle and DNA repair. And this will be important for the next slides. So in order to be able to validate these findings, we had to make use of another data set of samples. In this case, our lab has a resource where we have genetic expression of 71 cell lines that are patient-derived cell lines that we have established throughout the years. And so basically, when there's a patient and they're going to do a tumor resection, we get a piece of the tissue. We can dissociate and generate a single cell suspension that we can then culture and establish what we call a GBM sphere culture. These cells can be then used for different analysis in vitro and eventually implantation into mice after we have done any genetic or pharmaceutical intervention into the cells. And so I did a similar analysis in these samples. And in this case, I generated 2 different clusters using both a canonical and oncogenic pathways. Again, I was comparing each sample to the average of all the samples and then clustering them based on the enrichment profile of each sample. And you can see here both of the clusters. So going back to the approach, we've gotten to this step. And so the next step is to see what are the common elements for each of the clusters, and then using ingenuity pathway to try to identify targets that we could use to validate our approach. So this is sorry. So I did this validation, and if you guys are not familiar with ingenuity, what it does is it looks for connections in the literature. So you can provide a list of genes based on, in this case, it was based on how they contribute to each of the particular clusters. And this list, it looks in the literature to see if there's connections between the members of the genes that you have inputted into the software, and provides not only connections, but also you can look at a tool that is called upstream regulators. And so if there's something upstream that could connect all the members in the gene list. And so what I'm showing here is a similar study where I compare the enrichment profiles of all the samples, but instead of using the gene sets in the collections that are from the software, I use gene lists that I generated based on their contributions to each particular cluster. And again, I get two distinct clusters in this data set. And next to it, where you see is from the upstream regulator tool from Ingenuity, what are the upstream genes or proteins that seem to be activated or inhibited in each of these gene lists. So if there's enrichment for a particular gene list here, then the prediction is that these molecules are either going to be activated or inhibited. And so I was very interested to see that for these two different gene lists, the software predicted E2F1 to be activated in both of them. And it's more significant in the sense that if you see here, this gene list came from the TCGA analysis, so from the TCGA dataset, and this one came from the Neurosphere dataset. So even from two different datasets, we get to a similar common upstream regulator. So I just did a little bit of validation in the sense that we know that E2F1 is associated with a cell cycle. And so I took some of the genes that are associated with cell cycle, and then I look at their enrichment in all of the samples, and then I label the samples based on whether they are enriched or not for E2F1. And you can see that the samples that have higher scores for cell cycle gene sets are also the samples that are predicted to have E2F1 activated or enriched. And the same is true when I did this using targets of E2F1, where there's a higher enrichment in E2F1 targets in the samples where we predicted to have E2F1 enriched. So this was a nice validation of our findings at this level. I also went back to the TCGA and reanalyze everything using just these gene lists. And you can see that again, these 7 gene lists are able to cluster the samples into 3 distinct clusters. And the arrows represent how each one is contributing to each of the particular clusters. But again, we don't have access to the TCGA samples. And so in order to validate our findings, we are using the patient-derived lines that we have access to. Now, before I go into the validation, the other question that we had is, okay, we've identified these gene lists and we have some upstream regulators for each one. Can we relate that to intratumoral heterogeneity? And so this is from the original paper that described the presence of different molecular subtypes in each of 5 samples that they use in this study. This is how the proportions of each one are present in each of the samples. And so we wanted to know if there are specific pathways, if there's a dominant pathway, in this case, if there's an enrichment for one particular one of the genes that we have identified in each one of the samples, and whether we can associate this to cell cycle or stemness. And so we did analysis in these same samples that you can see here, and we were trying to correlate the different gene lists that we had generated with, in this case, cell cycle signatures. And you can see that the 2 that predicted E2F1 to be the upstream regulator are the ones that have a very positive correlation with cell cycle, whereas the others don't. And there's this one that had interferon gamma and NF-kappa B as upstream regulators that show negative correlation with cell cycle signatures. We also did the same analysis, but instead of cell cycle, we use it with the stemness signatures. And there's one notable one where EGFR is an upstream regulator, and we see a negative correlation between EGFR expression or EGFR activation and stemness, which is interesting because EGFR is one of the most mutated genes in glioblastoma. Keeping with the single cell studies, there was another study in a lot more samples published a few years ago, where they not identified the classical molecular subtypes, classical mesenchymal and proneural, but rather they were able to tie together cellular states to mutations and also sort of associate those with different populations that are present in the brain. And so they came up with OPC, like for oligodendrogenital progenitor cells, MPC, like for neural progenitor cells. This is astrocyte-like and mesenchymal-like. And so what they propose is that in each tumor, there's different proportions of these cells, but all of these are present in every tumor. And so the question was then, can we relate our gene lists to these signatures? And that's what is shown here. This, sorry for a busy graph, but there's no other way of showing all the associations at the same time. So there's three that we're mainly interested in, and you can see those have a square. So there's three that have a square on them, and those are interesting to us because they associate with some of the lines that were described in the cellular subtypes. So this one associates with OPC-likes and MPC-like cells. There's another one here that associates more with a mesenchymal subtype. And also, there was another report in GBM that showed that there's the presence of a rare tumor population, or sorry, there were tumor cells that associated with a rare population of cells that exists in the developing brain called outer radioglia that are mostly absent or during development, are not so present in adults. However, GBM cells are capable of expressing a signature that identify them as outer radioglia. And so we had another gene set that highly correlated with this newly described phenotype. Also here, I'm showing how or what the upstream regulators of each one are, and also whether they positively or negatively correlated with stemness or with a cell cycle. And so there's a lot to unpack still in these associations, and a lot that we can still study to see if there's more into this in terms of the gene list that we have identified, and whether we can generate some therapeutics with this. And so to finish this part of the talk, I'm just going to show how we validated these studies. So we focus on E2F1. As I mentioned, E2F1 is very much an integral part of the cell cycle process, as in shown in here. But E2F1 has also been described in a lot of different metabolic processes, such in thermogenesis, adipogenesis, and both like stimulatory and inhibitory effects. And so it's very surprising to anyone studying cancer to say that E2F1 can only be important for a subset of cells. Because if you think about it, E2F1 having such an important role, if you knock it down or you you inhibit this function, it's definitely gonna inhibit cell proliferation. And so we were interested in trying to show that we can predict which samples are actually gonna be sensitive to E2F1 inhibition. And so that's exactly what we did. We silenced E2F1 using silencing techniques, and we had 2 samples from the E2F1 activated clusters and 2 samples from the E2F1 non-activated cluster. And this is all based on the genes and enrichment analysis that we did. And you can see that knocking down E2F1, in this case, it decreased the sphere formation capacity of these cells, whereas in the non-activated cluster, it didn't change their sphere formation capacity. If you guys are not familiar with this, these are limiting dilution analyses where we played different numbers of cells in a 96-well plate in a limiting dilution manner. So if you start with 100, then you go to 50, 25, 12. And after 7 days, you count how many of the wells were able to form spheres. And based on that, you can generate these graphs and calculate a stem cell frequency. I also have proliferation data where the same trend was shown, but I'm not showing that because of space. And then we moved to in vivo, where we implanted these tumor cells that also were expressing luciferase, so that we can image these mice in vivo to see the tumor growth through time. And you can see 301 is from the activated cluster. Knocking down E2F1 shows a reduced signal of luminescence, meaning the growth of the tumor in this case, whereas 408 doesn't show a difference. And we measure these things through time, and you can see a clear differential or a statistically significant difference between the control and the E2F1 knockdown group. And this was not true for 408, which was predicted to not have E2F1 activated. And in terms of survival, it did improve survival of knocking down E2F1, did improve survival in the 301 group, but it was not significant in the 408. And so with this, I'll just summarize the first project. So we were able to generate this genes and enrichment-based analysis to try to come up with new targets that have clinical significance. And we uncover differential sensitivity to E2F1 inhibition. Importantly, we were able to predict which samples were going to be sensitive to E2F1 inhibition. And we also described cell populations that have or populations that have enrichment for different gene lists, and we link them to different cell identity and molecular states that have been described in GBM. Now for the second project, like I said, it's common in the sense that we are using a pathway-based approach, but in this case, we're using mass cytometry. So for people unfamiliar with mass cytometry, it couples flow cytometry with mass spectrometry. And so basically, we have these lines that we have established from patients that we can then generate a single cell suspension from. And then we can label this with antibodies, just like you would do for flow cytometry. But the difference here is that the antibodies are conjugated to metals. And these heavy metals are not present usually in live cells or animal cells. And then once you've labeled the cells, you put them through the machine, and the machine is gonna ionize each cell and gonna release the metals that are attached to each cell. And just like flow cytometry, it goes in a cell-by-cell basis. And so after you read the metals that are attached to each cell, you generate sort of like a snapshot of each cell. which then you can put together based on the profile for each of these cells, and you can generate clusters again. And this is just an example of that. So for this analysis, we generated a panel based on dysregulated pathways that have been described in GBM, and also included different stem cell markers and markers that have been associated with GBM, like I mentioned in the introduction, like CD133 or CD44 or CD15 that have been proposed as markers for cancer stem cells. It is also important to think about the tumor microenvironment when thinking about what we can do with mass cytometry. So there's been 3 different niches that have been described in GBM. There's the invasive niche, where one of the properties of GBM is that it's highly infiltrative, so it doesn't grow encapsulated. It's able to have this invasive niche. And so the cells that are present in this niche are not the same as the cells that are in the core of the tumor. In the core of the tumor, there's this hypoxic niche, where the cells that do not have access to oxygen or nutrients, but they are still able to survive by expressing different markers. There's been a lot of study in this area. And then there's the angiogenic niche, where there's cells that are highly interactive with endothelial cells, or even cells that are themselves becoming endothelial-like, but that are from tumor origin. And of course, there's the presence of microglial cells and astrocytes, and also immune cells that infiltrate the tumor. So just to have that as a background. Now, the first data set that I analyzed using this panel was in collaboration with Ichiro Nakano. And what he did was he's a neurosurgeon. So every time he was dissecting a tumor, he will generate different clones from different areas of the tumor. So he would cut pieces of the core and the edge of the tumor, and he will plate the single cell suspension in very, very low numbers to generate these clones from different regions. And then there's been a lot of studies in terms of gene expression and other sequencing methods, trying to look at differences between core and edge samples. But in this case, I was mostly interested in taking these clones from the core and from the edge, and to see if I could find any differences in terms of signaling and stem cell marker expression using mass cytometry. So this is what I found from these samples. I generated 12 clusters based on the expression profiles of all the cells in these clones, where there were mainly 4 different clusters that made up about 80% of the cells, and there were other clusters that were that represented about 15-20% of the cells. So this is just an overview. You have different markers and the expression of all of the markers in each of the clusters that we identified. Now, if we look at the abundance of these clusters, comparing core in red versus edge in green, even with these low numbers, you can see that there's significant differences, which are the ones that are labeled in red and green, between core and edge. And if we look at how the stem cell markers are expressed in each of these clusters, then we see that they have in common the fact that SOX2 is highly expressed in the edge clones compared with core, whereas A2B5 is actually higher in the core clones in these clusters that were significant. But this is just a first approach into trying to characterize these clusters. The main downside of doing this into trying to identify different tumor populations is the fact that we don't have any knowledge of which of these markers is going to identify a different tumor subpopulation or not. People have been using CyTOF mainly in immune cells where known markers are available, and so you get very distinct populations. But in the case of tumor cells, we have to try to tease out which markers might be important or not. So in order to circumvent that, I found this software that was developed by people in Vanderbilt, where they came up with this marker enrichment modeling, or MEM, where they would compare the expression of all the markers in all the clusters that they identify and gave a MEM score. And the MEM score, the higher it is, or the more positive, it tells you how important is that marker for identifying that particular cluster compared versus the other clusters. And it doesn't mean that high MEM score means high protein expression, because if you can see, this is for immune cells. All the cells down here, it's protein expression. So down here, all the cells express high levels of CD45. And so CD45 is obviously not going to be a good marker for differentiating between the different clusters. And so it has a score of 0. But for other clusters that are, or for other immune populations that are highly specified, there are some markers that are, for instance, only B cells are going to express CD19, like you see here. And so CD19 has a high score, has a high MEM score. So sometimes it does correlate with expression, but only when it's like very highly specialized. But if all the cells express the same marker, then it's not going to be very useful. And so with that in mind, I generated these MEM labels using the software for our clusters. And what I found was that there were a few clusters that separated from the other clusters. And what these had in common was they had high MEM scores for SOX2 nesting and CD44, which was not true for the ones down here. And the other important thing of these MEM labels is the fact that they are computer generated, and they are also computer readable, in the sense that if you have labels from different data sets, you can put them together and see if they are able to be associated. So just keep that in mind for the next few steps. Now, we didn't just want to study differential populations that came from different places in the tumor, but we also wanted to try to associate this to a clinical setting. And so in collaboration with colleagues in Cleveland, they were running this phase 0, phase 1 clinical trial. Well, they were giving patients before surgery a low dose of this drug, which is a 5-FU prodrug that they were trying to use for the eradication of MDSC populations, which promote immunocompromised state in a tumor microenvironment. And so they treated patients before tumor resection, and then they collected this tissue and they sent it to me. And the first goal was to analyze the immune populations in these samples, which was part of a study that we published. But I also analyzed the same samples with my signaling panel. And so the idea was then to try to associate the abundance of different tumor populations that we could identify with the abundance of immune populations that we also did in the same samples. And so in this case, I identified 13 clusters. Again, there's 5 or 6 that make up about 75 or 80 of the whole tumor, and then there's some other minor populations. There are some that even at these low numbers are significant. Between ND means newly diagnosed, and these are recurrent samples. I also did a comparison between samples that were treated with the drug and compared with recurrent samples that were not treated. And we could see a shift that was significant in some of these samples. But what we really wanted to do, I'm sorry, and this is the immune part that I did for these samples that was published a couple years ago. And we identify a number of immune populations. And like I mentioned before, when you are dealing with immune cells, mass cytometry is great at identifying and very nicely having these distinct tumor or immune populations. And you do see differences between the treated ones and the recurrent samples in terms of the abundance of some of immune populations that are important for the immune response against the tumor. So what we wanted to do was to see if the clusters that we identified were actually associated with some of the immune populations that we later found. And the answer is that some of the clusters that we identified and that were differentially expressed between newly diagnosed and recurrent were associated, or their abundance was associated with the abundance of immune populations that are important for the immune response, such as NK cells and microglia and CD8 effector cells. Whereas there were another group of clusters that were associated with some immune populations that are considered detrimental for the immune response, such as macrophages and monocytes and some NK cells that are not very effective at responding against the tumor. So as I mentioned, we want to try to associate these two data sets, and the way of doing that was using MEM. So the marker enrichment model has been proven to be able to associate similar populations. So in this case, we're looking at T cells and B cells that were labeled in very different experiments. Some come from peripheral blood, some come from bone marrow, some are done using fluorescence, others are done with flow cytometry and mass spectrometry here. So what they were able to show is that by putting these labels together, they were able to distinguish between two different cell types, and that the higher this root mean square difference was, the more likely that these populations associate. And so I extrapolated this into our work. And what I was trying to do was to see if some of the tumor populations that I identified in the CORE-NH clones were similar to some of the tumor populations that I found in the clinical data set. And so what you see here in black is the clinical clusters that were identified for the clinical data set. And in green, you get the clone clusters from the core and edge. And so we were able to find that there's some associations that are highly significant, and extrapolating the first MEM score labeling that I did, we found that in clone 7, which is highly abundant in the edge clones, where there's higher scores for SOX2CD44 and nesting, that one can be associated with different clusters in the clinical dataset. And the same is true for some of the clones or some of the clusters that were present in the clones dataset that had lower scores for SOX2CD44 and nesting. And so we started thinking at SOX2CD44 and nesting as a population that we can try to identify and isolate. I'm going to skip this. It's the same analysis, but using signaling markers instead of stem cell markers. So I went back to the clinical data set and just try to gauge cells that are triple positive for these markers that I identified. And we were able to show that there's more of this triple positive population present in the recurrent samples compared to the newly diagnosed, and also compared to the treated samples in this case. In terms of signaling, and again, this is limited to what we have in our panel, we also saw that there's very big differences between the triple positive, which are on the left for each of these comparisons, versus the triple negative, which are in the right. And some of these markers have various implications in terms of signaling and survival. We were also able to show that just as the clinical clusters, the triple negative and the triple positive were able to associate with different immune populations, where the triple positive associates with these populations that are detrimental for the immune response, like monocytes and macrophages that have been recently described in the literature, whereas the triple negative are associated with C effectors and NK cells and microglia, which appear to be pro-immune response in the tumor microenvironment. I'm just going to skip that. All right, so just to finish, some future directions in this project. We've done all of these using samples that we had access to, and it all in vitro. And so thinking about how to validate this in vivo, we have a collaborator at Cedars-Sinai that have been developing genetically engineered mouse models, but instead of making use of a lot of crossing different mice with different mutations and waiting for the tumors to appear, they've been able to do in vivo electroporation of different genes in what they call a mosaic analysis with dual recombinases. It's very interesting work if you guys want to take a look. But the original work was done in pediatric gliomas as well as ependymomas, and they were able to generate a lot of these mouse models. But lately, they've been working into adult GBM model, and it's mainly based on EGFRB3, which is a known mutation amplification in a subset of GBMs. And so what they did was they generated this mouse model, and then they harvested the tumors, and then they cut it, and instead of keeping a cell line from them, they generated what they call MGOs that are mouse GBOs for glioblastoma organoids. So they kept them in this way to try to preserve the heterogeneity, because when you culture something as a line, it loses some of the heterogeneity. So they were nice enough to send me some of these, which they was able to grow and dissociate and implant into mice. And the idea was to see if we could observe the same populations and the same clusters that I identify in the human side. So this is just preliminary data that I've gathered recently. I have 5 different mice that I've analyzed here with a signaling panel. These are how the expression markers are present in each one. I was able to see different expressions of different levels of expression of nestin CD44 and SOX2 in some of the clusters. And I've also generated MEM scores for the clusters that I identified. And it doesn't exactly copy what I saw in the clones data set. But I did see some populations that have high scores for CD44 and nestin. And the other main point about using this mouse model was to try to see if we could also identify different immune populations that way that then we can explore on after treating the cells with radiation or chemotherapy and see if we can see shifts in the abundance of these populations. And so again, this is preliminary, and I was able to identify 10 different immune populations in these mice. These are the abundances here. And so the next steps are to try to treat these mice with different drugs and maybe radiation, and see if the shift in some of the tumor population associates with a shift in the different immune populations. And that's based on the previous work that we've done. Also importantly, in this panel, we have a lot of markers for exhaustion. So that's a topic that has been in the literature lately. The fact that T cells and NK cells are present in the microenvironment, but they can become exhausted. And so they don't respond and they don't generate the immune response against the tumor. And so some of these markers that you see here are very, very interesting, and we're going to be studying them later on. And finally, we have another study from the lab that we just deposited into bioRxiv that is looking at similar to the CORA-NETCH. This study focuses on spatial heterogeneity, but the advantage here is that they took samples from different areas of the tumor, and they did, in this case, single nuclei RNA-seq, and we have access to this data. And so we've been trying to see if the existence of these triple positive cells is different in the different locations. And so far in 3 recurrent GBMs, we don't see a consistent trend, but it will be interesting to compare recurrent GBMs to primary GBMs, because I think that's where we will see a bigger difference between the existence of this triple positive population. It is true that if we look at the clinical outcome of these patients, a higher abundance of triple positive cells appears to be significant in terms of patient survival. But again, it's a very small cohort, so we're not making any conclusions yet. And finally, we did look at differentially expressed genes in these triple positive versus non-triple positive in each of these 3 tumors. These are the numbers of differentially expressed genes in each of them. And then there were about 195 shared differentially expressed genes. And just using genes and enrichment, these are the gene ontology terms that are mostly significant. These one more intense are the ones that are highly significant. So synapse organization, axon development, and regulation of neuron projections appear to be important in the triple positive populations, but we're going to continue working on this in trying to identify a marker or a biomarker that then we can use for our future studies. And so to summarize this second project, we're focusing on the different regions of the tumor to try to identify different potential subpopulations in the same tumor that we can then correlate with the abundance of immune populations in those same tumors and try to see which populations are important for the immune response. And that might be important also for resistance to therapy. So future directions, like I say, we would like to start looking at CyTOF directly using enhancing versus non-enhancing regions of the tumor and doing CyTOF directly in those, seeing if these populations are more abundant if recurrence, comparing primary versus recurrent samples. When looking at the triple positive population, we've been trying to isolate this. We actually were using fluorescent reporters recently, and we've been able to identify and isolate this population, and we're starting to do some studies with that. We're mainly interested to see if in response to radiation and chemotherapy, this population becomes important or not for the tumor cells. And the short answer so far is yes. And then finally, we are very interested in tumor immune populations or tumor immune interactions that could be before or after immunotherapy. And from the gene expression analysis from the single nuclei RNA-seq, we're starting to look at those interactions as well. So finally, I would like to acknowledge the members of the Kornblum Lab and Harley Kornblum, as well as our collaborators at UCLA and elsewhere, and also the NINDS, and I also make a research foundation for their funding, as well as the UC postdoctoral fellowship program that gave me support for 2 years. Thank you.
Okay, thank you so much. This was a wonderful talk. So we have time for one question. Everybody? Anybody? No questions? Because I mean, many people left already, but I have a question that is the question. Okay. So I see that you have been able to classify all these cells in many subgroups, right? So the question is how you adapt all this knowledge that you generated to treatment.
Mm-hmm. Yeah, that's something that I've always had some reservations with some of the papers that were published in the field throughout the years, because just coming up with different classifications of the tumor without any downstream applications, then it kind of doesn't serve any purpose. So that's why in the first project, rather than focusing on looking at differences between the clusters and whether we can keep showing differential expression or identifying another biomarker or something like that, I focus on the targets that we can predict to be activated within the different samples, and trying to validate our approach that way. And in terms of the mass cytometry, what I envision is trying to identify a potential subpopulation. It might not be a distinct subpopulation, but something that is relevant functionally for the biology of the tumor, and more importantly, something that is associated with the abundance or the presence of certain immune populations, and targeting it that way, rather than trying to eliminate it from the tumor or just focusing on characterizing this tumor population.
And how long do you envision until we can do that?
That's a good question.
You don't know, right?
I don't know.
Many years, I'm guessing.
Depends on how much funding you get, I guess. I don't know.
I guess. Well, thank you so much, Alvaro. It was a pleasure to have you. and we will send you the recording of the talk. Okay.
Okay.
Thank you so much.
Thanks.